So gymnosperms are the oldest surviving group of seed plants after the gymnosperms. They're coming out 360 to 340 million years ago. Whereas the flowering plants, and the overall term for angiosperms, people refer to them as the Anthophyta. The Anthophyta are much later. They do not really appear. We cannot recognize them as such. It takes a while to get a fossil flower. The oldest ones are probably about 120 to 122 million years ago, which actually puts them in the Cretaceous, when the various continents are separating from each other and then likewise banging into each other and setting up new mountain ranges like the Himalayas. Some people will argue that, well, the flowering plants really probably appear at the end of the Jurassic. That's so where things are closer together and it's easier for them to move. But the fossils from that period, while they show certain angiosperm-like characters, you're still not getting the closed ovary, which is what makes an angiosperm an angiosperm. It may just be a matter of time before we find them. It's certainly a lot better that when compared to when I went off to college in the woebegone days of the early 70s, in which the earliest fossils were about 65 million years ago. They seem to appear around what we think of as the age of mammals. In fact, we now understand they're much, much older than that. But we can now say that between the gymnosperms and the angiosperms, there are at least 750,000 living species of seed plants on this planet. There are more seed plants than there are plants that lack seed. And the modern forests are dominated by seed plants growing as trees. The major advancement over seedless vascular plants is that the gametophyte is now dependent on the sporophyte, and you don't need that wet stage in which sperm has to leave the gametophyte and go swimming around and looking for an egg. This means that that seed plants are dispersing fully formed embryos in containers that are resistant to the elements, but they're also packaging their seeds with a source of food. So during this dormancy period, they are respirating, but it's usually a form of anaerobic respiration. So all seed plants are technically heterosporous, producing distinct male and female gametophytes in the form of a pollen grain versus that of an embryo sac. In regards to diversity, though, while we still have gymnosperms, 97% of all the seed plants on this planet living today are angiosperms. So if we check the other handout and simply go looking at the second page, we notice that in seeds there are things that they all tend to share, and then there are things that you only find in some of the seeds and not in the others. We should expect three basic components of a seed. The first of this is the coat or testa. These are mature integuments, and they are basically protective. They stop things from getting into it as the embryo matures. And that's the second thing. We expect to see an embryo inside, which is the beginning, which is what happens after you fertilize the egg. Now, you should see some food tissue also. This is a source of food and gas exchange, but this tissue can change radically as the seed matures. In some cases, it gets gobbled up even before the seed sprouts. So, if we look at seed coats, first of all, what are we talking about? We're talking about these integuments that lose water at maturation. Seeds are usually dehydrated prior to dispersal. When a seed is released by the parent plant, only 15 to 20% of its body weight is water. So if we look at these structures, we are to attach this to a fruit wall. And here we have a placenta. We find that the majority of at least flowering plants will have these two basic integuments that are going to cover the interior embryo sac. And that upon fertilization of the egg, we are going to expect that the integuments will change. They may get larger initially, they may take on certain colors, they may even get hairy or scaly at certain points, but they eventually lose water and become highly protective. But when a seed is released by the parent, we're going to find that virtually all seeds, if you look carefully enough, are going to have two rounded scars on them. The first of these, which is the largest and easiest to see, is referred to as the hilum. This is where the seed was attached by means of placental tissue. In fact, some seeds take the placenta with them. The placenta ripens along with it, and you can see some of these little seeds with a fleshy structure, often colorful and smelling quite nicely, attached to the seed. The hilum in this case often fills with amino acids and lipids and triglycerides and becomes very attractive to certain animals, which take the seed away, eat the hilum, and leave the seed somewhere to germinate. Ants are particularly interested in seeds that have a fleshy placenta. A fleshy placenta can be called a caruncle. And since ants can be very messy housekeepers, this is great for the seeds because the seeds, once the caruncle is removed, go into the ant's garbage pit, and that is where they sprout. But there is another structure here, a small pore, which is what? You get two scars. One's big, that's the hilum, that's placenta, and the other one is what? The other one, this other pore, That's the micropyle. That's all that's left of it. Now think of it. Comparatively speaking, when you think of it, a hilum, which was formerly attached to a placenta in an ovary, is comparable to our own belly buttons. But there's one thing animals don't have. We don't carry around the pore by which our father's sperm entered into the egg. Now, do we? So, let's move on. Yeah, I think we need to have a little break first, and I'll put a few things up. And those of you who didn't pick up the handouts, might be a good time to do so. Everyone had a chance to sign the... Oh, people are still at it. We'll move along then. We've had a look at the seed coat. Let's look at the embryo. Embryos of plants, mature seed embryos of plants, should be composed of three parts. There should be three parts. Some get by with only two. But an embryo should consist of an epicotyl and a hypocotyl. The epicotyl is the shoot. The epicotyl is what will eventually escape and produce stems and leaves. So we expect that the epicotyl will be covered. It will be clasped by these tiny microscopic leaves known as plumules. They will eventually expand into the first true foliage leaves. At the bottom, we should see a hypocotyl. This, of course, is the embryonic root. It usually has a swollen tip referred to as the radicle, and then we would expect, even at this early age, it is covered by a series of dead cells known as the root cap. You can think of it coming out, pushing itself into the soil, and of course you need some sort of a helmet, some sort of a protective device so that it isn't ripped to shreds, because we're going to find that in both epicotyls and hypocotyls, there will be centers of mitosis that we refer to as meristems. More on this later, and it's important to realize at this stage they are both primary meristems inside, producing primary growth, which means length is more important than width. Now, the next feature is where we first see a number of these seeds diverging, and that is cotyledons. Cotyledons are embryonic leaf-like structures that often have a variety of jobs, and most of the ones that you're going to see on Wednesday and Thursday, their primary job is early photosynthesis. They get the seedling done, make some sugar before the plumules have a chance to really take over. But you'll see that there are a great deal of differences between the, first of all, gymnosperms and angiosperms. Gymnosperms tend to produce quite a few cotyledons. They can produce as many as 8. Whereas if we take the flowering plants and subdivide them into monocotyledons and eudicotyledons, these are the major classes within the phylum Anthophyta, we see that monocots usually produce only one cotyledon, and very often it's not photosynthetic. It serves as sort of a stomach. It is actually serving as a digestive organ that is going to help break down the stored food. It's not always photosynthetic. Whereas in the eudicotyledons, we expect that they are going to produce 2 cotyledons, each one flanking the epicotyl, as you can see. The function and shape of cotyledons, as I said, tends to differ between monocots and eudicots. In the eudicots, the cotyledons are usually photosynthetic. They will make food. They may even contain food. When you think about it, every time you eat a bean or a pea, most of the nourishment is from the cotyledons. They've already absorbed the stored food during the maturation process. So cotyledons may vary in thickness in eudicotyledons. When they are thin and flexible, it usually means that there's lots of stored food in the seed in the form of what we would call endosperm. More on that in a moment. When they are thick, succulent, and hard, we expect that they've already digested the endosperm and they're in fact containing it within the cotyledons. And I said in monocots we expect that the cotyledons are not photosynthetic at all. They're acting as a stomach, accumulating and transferring endosperm sugars. as they are broken down during the act of germination. That is, the food is broken down by enzymes during the act of germination. In some grasses, and all grasses are monocots, the cotyledons never leave the interior of the hard seed. They're just a hard little stomach as the epicotyl and hypocotyl break out of the seed. They're rather hard and stony, and when you pop corn, that little brown scab in the center of a popcorn is in fact the single cotyledon, and it's referred to as a scutellum. Looking then at food tissues, they are going to differ cytogenetically in seed plants. In gymnosperms, the food tissue, as you can see, is provided exclusively by the female parent following meiosis. So in gymnosperms, food tissue is always haploid. This means it develops much more slowly than the embryo would, and one of the reasons it takes so long for gymnosperms to mature, frequently just getting them out of the seed, is because the cone will hang on the parent, slowly picking up food into its haploid tissue from anywhere from a year to 18 months. It's that slow a process. Angiosperms, on the other hand, have this amazing endosperm, which in fact is triploid. I will have more to say about this in weeks to come. But if we look at most embryo sacs, here is your egg. In the center of the embryo sac, we normally see these two cells mushed together. They're haploid, but they're compressed together, and these are known as the polar cells. It means that when a pollen tube delivers its sperm, it's not delivering one sperm to an embryo sac in an amphiphyte, it's delivering two. One will unite with the egg, and this gives you the diploid embryo, but one actually unites with the polars, and this gives you a triploid endosperm. Triploid mitosis is fast, development is fast. We find that in the angiosperms, the formation of an endosperm is often accomplished in a period of just a couple of weeks. This means that the endosperm inside of an angiosperm seed is going to develop as rapidly as that of the embryo. Now, in some angiosperms, endosperm never forms. The seeds are photosynthetic and are ready to photosynthesize before they germinate. As in the case of rhododendron seeds, we see the seeds of rhododendron. They have a transparent seed coat, and they have little green embryos inside, and they're all ready to go almost as soon as they hit the forest floor. And then you get others, like the case of the orchids, which I've already mentioned, of which there's more than 20,000 species. Virtually all of them do not produce endosperm, but they have to go very quickly. Once they fall to the ground, they have to very quickly join up with a fungus that makes endomycorrhiza. when you think about it, it's the fungus that is feeding the orchid, often for a period of several years until they develop their first leaves. If we look at embryonic development in seed plants, after fertilization, the zygote starts dividing and enters what we would call a proembryonic phase. Here, once again, this is an early period of development, and it is pretty much identical in monocots as well as in eudicots, which we will concentrate on at the present time. What should we expect in the early development of a seed embryo? What makes something a proembryo? What we should expect to see here is that it will consist of two parts. You're thinking of something developing inside of this ovule at this point. We should expect to see, first of all, something that looks like a chain ending in a big fat basal cell. This chain is called the suspensor. The second part of this will be the proembryo, a multicellular proembryo. Which will develop into the full embryo that I've talked about with an epicotyl and a hypocotyl. But at this point, there is a distinction between monocots and eudicots. In the eudicots, we expect the proembryo will develop into a little heart-shaped structure. The two lobes here will become the cotyledons. We have the little epicotyl here, and the bottom half will become the hypocotyl. In monocots, on the other hand, the proembryo simply gets longer and more torpedo-shaped. At most, what you get on one side is the formation of a little notch, which you can see. So the top part is your single cotyledon. Here is your epicotyl, and here is your hypocotyl still attached to its suspensor, and you'll be able to see the difference in lab this week. What you do with the suspensor also seems to show a difference between monocots and eudicots. In angiosperms, the suspensor may make proteins for rapid growth of the proembryo, or the suspensor acts as a pipe or conduit so that the proembryo absorbs nutrients from the developing endosperm. Not in gymnosperms. In gymnosperms, the suspensor does nothing more than push the proembryo into the haploid nutritive tissue. Now, this is why the seeds of some eudicots lack endosperm and have these big, lumpy, hard, fleshy cotyledons. What's happened then in the case of some of the eudicots is that the basal cell has hooked them up to the triploid endosperm, and the nutrients in the endosperm goes into building the proembryo, which means you tend to produce these embryos with these absolutely huge, fleshy cotyledons. And if you don't believe me, the next time you have guacamole, save the seed, let it germinate. When the little epicotyl comes out, you can open it, and you can see that those are just two big cotyledons that have been feeding the little epicotyl. Which takes us then into this matter of dormancy and germination. The embryo of a seed is mature once it forms all its organs and crushes the suspensor. When this occurs, one of two things can happen. In a few cases, the embryonic root or radicle continues to grow until it breaks through the seed wall. The seed is released from its parent and it grows independently immediately. This is a relatively rare occurrence. When you think of what's going on here, the seed is still attached to the parent. It may even have a little bit of a fruit wall around it. And it's producing a little dagger-like hypocotyl, and then breaks off with the parent and probably will fall into soft sand or mud. A number of trees that live in estuary areas in the tropics or in Florida that we think of as mangroves do this. It's a way of pre-planting your seed in an area that has a tide going in and out. You can't afford to wait. You got to get the thing established in the mud flat, or it's going to be carried out to sea, right? We also have this group of marine plants in tidal areas known as seed grasses, and they're taking it even more seriously. Once the hypocotyl comes out, the hypocotyl starts branching into these little claw-like structures that allow the seed to grasp the sandy bottom. But this is rare. Most seeds are not ready for germination as soon as they mature. In most plants, the seed will then lose again between 15 to 20% of its body weight in water, and it enters a period of arrested development we call dormancy. In dormancy, the life of the seed hasn't stopped, it's just entered a slower phase. Why? The seeds of many plants require a slow, dormant phase to achieve enzymatic maturity, what we would call after ripening. This explains why in the fall some people pick fruits off of a tree, clean off the fruit flesh, stick them in a pot, and say, oh, I have a little tree growing on my windowsill over winter. I'll give it a head start, and nothing happens. For a number of our trees here in autumn, what they require is a period of after ripening. Their seeds are anatomically correct, they have all the parts, but they haven't started making certain enzymes yet. For that, they need a moist, low temperature for a period of weeks. So that's why they only start sprouting in the spring. Dormancy also, the second feature, is dormancy gets the seed through bad times. The seed stays in the slow phase until environmental conditions ensure peak survival. Consequently, there's a tremendous difference between the act of germination in a rainforest versus a desert. Trees in a rainforest drop their seed, or the seed is eaten by an animal and pooped out. They land on a wet surface soil and they immediately start growing. There is really not much in the way of a dormancy period. In deserts, on the other hand, where rain is is, shall we say, infrequent or then highly seasonal, seeds can stay there for a period of years. In fact, these seeds in their coats have these tannin molecules which are anti-germinants. They're not waiting for just some moisture, they're waiting to be thoroughly soaked. They're waiting for puddles to form. At that point, the anti-germinants leach out. It means the soil will stay wet for a period of weeks, and that is only when these seeds begin to germinate. Dormancy is halted by up to 4 environmental factors. In order of importance, they are water, oxygen, remember we have to go from an anaerobic to an aerobic state, temperature, and then in rather rare cases, light. Most seeds, as we know, at least most of our crops that we depend on, most seeds break dormancy following rehydration. Water enters through the micropyle or the hilum, or you have some seeds that just have a very porous or loose seed coat. What should happen then is that with water entering, the embryo tissues rehydrate, they swell, and then the seed coat ruptures or fragments. A lot of seeds have got this all built in, all taken care of. If you look at them, as you will in lab, we see that there are pre-made sutures on the seed coat. These are weak points, and as the cotyledons in particular fill up with water and start pushing outward, the seed coat splits naturally along these pre-existing sutures. In other cases, for example, like lima beans, as the cotyledons get bigger, the seed coat just fragments into a bunch of little scales and falls off. Once this occurs, of course, once the seed coat has ruptured, you have an invasion of oxygen to the embryo, so cellular respiration changes from anaerobic to aerobic. But there is this non-germination that can occur. People are just banging their heads against the wall. I can't get the seed to germinate. That's because other environmental pressures may control rates of germination. We've already looked at this matter of after ripening, in which the embryo is structurally complete but enzymatically immature. This is where, as I said, temperatures become important, particularly low temperatures. We tend to associate this with plants that drop their seeds in the fall and do not allow germination until warming conditions in the spring. Other seeds have impermeable coats. That's the end of that. There's no porosity, not even through the hilum or the micropyle. You require other forces to break through. This can be a case of slow erosion or mechanical cracking. In certain cases with alpine plants, you need a freezing period, freeze, defrost, acting on the seed coat. A large number of animals will have an effect on certain seed coats, birds with gizzards grinding it down, certain stomach acids, certain seeds must pass through the body of an animal before they are likely to germinate. And even fire in certain parts of the world. There are cyclical fires. We can think of certain floras in certain parts of the world, like the chaparral around Los Angeles as a flammable flora, or in certain parts of South Africa at the Cape of Good Hope and a lot of Australia. These are cyclical fires that blister the seed coat, which then pops, and then only then will water get to it. And it can be very dramatic. The seeds in these flammable floras wind up getting covered by debris year after year, twigs and leaves full of essential oils. A fire goes through, they are insulated a bit, but this burns away. The seed gets charred and it pops. And I've already mentioned the case of anti-germinants in various seed coats, particularly in desert flora. This is usually a buildup of tannins which have to be leached out. With germination, when we're talking about the embryo escaping from the seed coat, there's up to about 9 phases of what's going to go on, but we will save that for Thursday. Could I have the sign in when you're finished with it? Sign in, please. Who's got it? Oh. Sign in.